Good day, ladies and gentlemen, and welcome to the Adverse Outcome Pathways, Emerging Regulatory Requirements, and New In Vitro Models. All lines have been placed on listen-only mode, and the floor will be open for questions and comments following the presentation. If you should require assistance throughout the conference, please press *0 on your telephone keypad to reach live operator. And if you'd like to ask a question, simply type it into the chat box on the lower left-hand side of your screen and click the send button. And at this time, it is my pleasure to turn the floor over to your host, Director of Commercial Strategy, Graham Dick. Sir, the floor is yours.
Thank you, Daniel. Hello, my name is Graham Dick. I lead Commercial Strategy at Qualyst Transporter Solutions. We're a company that provides in vitro modeling products and services to help our clients understand and predict how the liver will metabolize and clear compounds. Thank you for joining today's webinar titled Adverse Outcome Pathways, Emerging Regulatory Requirements, and New In Vitro Models. We are pleased that Professor Mathieu Vinken and Dr. Jim McKim have joined us to talk about this important topic in toxicology. I will provide brief introductions for Professor Vinken and Dr. McKim before turning the podium over to them. As Daniel mentioned, if you have questions for Matthew or Jim, please type them in the dialog box on your screen. And we'll reserve time at the end of the webinar to address your questions. Professor Mathieu Vinken is an associate professor at the Department of In Vitro Toxicology of the Free University Brussels, Belgium, and an invited professor at the Department of Pathology of the University of Sao Paulo. He holds a doctoral degree in vitro toxicology, is a European registered toxicologist, and a trained chemical risk assessor, and is president of the European Society of Toxicology in Vitro. His expertise lies in the area of mechanistic and in vitro toxicology, as well as in the field of experimental hepatology. Dr. McKim is the founder and CEO of IonTox, a company providing in vitro toxicity screening services, custom research and development, and consulting. Previously, he founded CTOX, a biotech company focused on in vitro strategies for predicting in vivo toxicity. He has over 25 years leading and advising research efforts as an assistant professor at the University of Colorado School of Medicine, team leader for biochemical toxicology at Dow Corning, and a science fellow within Pharmacia. Dr. McKim's key areas of research have focused on understanding how chemicals affect key pathways controlling tissue health. He holds a Doctor of Philosophy in Biochemical and Molecular Toxicology from Oregon State University, is the primary inventor on 4 patents, has numerous peer-reviewed manuscripts, book chapters, and review articles, and has been a diplomat of the American Board of Toxicology since 1995. Matthew, thank you for joining. I'll turn the podium over to you.
So, thank you very much. First of all, thank you very much for the invitation to present today. So, welcome everybody. So that would mean good morning to the people in the US and good afternoon, on the other side of the ocean where I am, so that's in Europe. So today, I would like to discuss and to demonstrate to you the use of these adverse outcome pathways, these AOPs, as tools to assess chemical-induced liver toxicity. Now, before doing so, I would like to set a little bit the scene, so to sketch a little bit of background, especially for people not really familiar with the concept of these AOPs. Well, I think that we are all aware of the fact that there is a tendency, specifically in the last few years, to turn more to mechanistic toxicology, so rather than just, like performing toxicology assays and producing these apical toxicological endpoints, without really wondering about the mechanistic basis that drives these endpoints. And the first concept that has been introduced in this regard is the mode of action that is defined as a series of key events along a biological pathway from the initial chemical interaction to the adverse outcome. Now, this mode of action was first introduced in the cancer field by the US EPA, but throughout the years it has also been used in order to cover non-cancer endpoints, which, for example, has been done by the International Life Sciences Institute. So basically, this mode of action was mainly, if not uniquely, used for the field of human health risk assessment. And this is a difference with these AOPs. An AOP, which has its roots in the field of ecotoxicology, but especially in the last few years, it has become applied also in the field of human health risk assessment. Now, there are two major milestones here. So first milestone can be considered the introduction of this report, the publication of this report, and I think all of you are familiar with this, which has been published by the U.S. National Academy of Sciences, in which toxicity pathways play a very important role. And as a matter of fact, a toxicity pathway is the conceptual basis, or the basis as such, for an AOP, which can be defined, as you can see here below, as a conceptual construct that portrays existing knowledge concerning the linkage between a direct molecular initiating event, an MIE, and an adverse outcome at the biological level that is relevant to the field of risk assessment. Now, a second milestone, if you will, at least in this context, was the publication of another document, which was issued on behalf of the OECD, the Organization for Economic Cooperation and Development, and that was a kind of a template on how to develop AOPs, and also at the same time some guidelines on how to assess, how to evaluate these AOPs. And actually, there is a new project or a new template upcoming. No, it's not a template, a new document, a kind of an update, if you will, of this first document that was published in 2012. And in the meanwhile, it also has been complemented with this AOP handbook. And actually, this will serve as the basis for the remainder of this presentation. So on this slide, you can see the link between a toxicity pathway, a mode of action, and an AOP, which is illustrated by means of an example taken from ecotoxicology, but you can also easily apply this, of course, to human toxicology. And you can immediately see here that a mode of action is more extensive compared to a toxicity pathway, and in turn an AOP is more elaborate, is a broader concept, compared to a mode of action, because it can start with exposure, it does not necessarily have to, and it may go all up to the population level. So this is how an AOP roughly looks like, and some people like to refer to this as the so-called macrostructure. And in essence, an AOP depends or relies on two so-called anchors. So the first anchor is the actual starting point of the AOP, which is called the molecular initiating event, the MIE. I will be coming back to this further on. And then the second anchor is the actual endpoint, so the apical toxicological endpoint or the adverse outcome as such. And the entire space between these two anchors, which you can see here, is filled with so-called key events, which are interlinked by means of these key event relationships. Now, if you then have a closer look to the AOP, the microstructure, if you will, you can see that indeed all of these key events, so all of these information blocks, are located at different levels of biological information, which is of course fully in line with the definition of an AOP, which was discussed a couple of slides ago. So for example, you can have effects at the cellular level, like gene activation, at the organ level, like altered physiology, at the organism level, like impaired development, and in the even most extreme cases, and that mainly applies to ecotoxicological AOPs, you can have also these effects at the population level, like for instance extinction of entire species. So in the next 3 or 4 slides, I would like to briefly discuss on how to develop an AOP. So this is also done according to guidance that has been published also on behalf of the OECD. Now there are 2 approaches here. So basically you can start from each of the 2 anchors. So this is called the top-down procedure or a bottom-up approach. Now the first step in the top-down procedure, or the second step in the bottom-up approach, if you will, is the identification of the AO, the adverse outcome, which is defined as a change in morphology or physiology of an organism or system that results in the impairment of the functional capacity, or at least the capacity to compensate for stress. And here again, as you can see, which is again complying with the definition of an AOP, this can be located at different levels of biological organization. Now this adverse outcome can refer to a systemic effect, for example reproductive toxicity, but it may also relate to a local effect, like for example skin irritation. Now the second step in the top-down procedure, or the first step in the bottom-up approach, if you will, is the identification of the other anchor, so the first anchor, the MIE, the molecular initiating event, which is defined as the initial point of chemical-biological interaction within the organism. Now, very, very important here is that you define the sites of action of the MIE, because you can imagine that the adverse outcome, so the actual toxicological endpoint, can be very, very different depending on where the MIE is located. And this specifically holds true for receptor-ligand interaction, which is a prototypical example of an MIE, and other examples are protein oxidation and DNA binding. So then the next step in AOP development is the identification of these key events, which are defined as a change in biological state that is both measurable, that's very important, and essential in the progression of a defined biological perturbation leading to the actual apical toxicological endpoint. So basically, as mentioned already before, the entire response matrix between the first anchor and the second anchor. As I will discuss further on, until a couple of years ago, the OECD also made a distinction here between these key events and intermediate steps. So key events, which are still valid, are measurable and are really essential for the AO to occur, whereas intermediate steps refer to information blocks, so in this response matrix, that were there, but that were not really essential for the adverse outcome as such to occur, and that were also not necessarily measurable. So the OECD gave up this distinction between a key event and intermediate steps. So everything now between the two anchors is called a key event. Now, I regret this a little bit because this can have some applications. Now, I will be coming back to this further on in this presentation. So there are no real restrictions with respect to the number of these key events, but they should make sense. So this means that you should have a very good understanding of the normal physiological pathways that they underlie. Now, there are many ways, of course, for the identification of these key events. The most obvious thing is going through all of the relevant current scientific literature, and this also explains why in AOP in general, it's quite a labor-intensive work, so to say. But in essence, you're not only supposed to go through literature, you should consider all of the data that are available. So, for example, animal data, in vitro data, in chemical data, in silico data, and especially in the last few years, we have all of these omics-based data, and this will be exemplified also further in the presentation. So then the next step in AOP development typically is the description of these key event relationships that define a direct relationship between two key events, or that facilitate the extrapolation of the state of the downstream key event from the upstream key event. Now, these key event relationships may refer to a direct linkage between a pair of key events that are adjacent in an AOP, or they can refer to an indirect linkage between a pair of key events for which the relationship is thought to run through another key event or a gap in current understanding. Now, the reality is that in most, I would say the vast majority of all of the AOPs that we have available today, that these key event relationships are rather of qualitative nature. Of course, if we want to use these AOPs, especially for regulatory purposes, then we should strive for quantitative kind of key event relationships. And these can be very diverse in nature. So, for example, these dose-response relationships, thresholds, points of departure, mathematical equations, or these biologically based computational models. And this is something that specifically applies to ecotoxicological AOPs, namely the fact that they might also be influenced to modulating factors. So that was the first stage, the AOP development. So after you came up with a new AOP, you need to evaluate this. And here again, the OECD has provided some guidance. So they propose two approaches that should be run in parallel. The first approach is a so-called weight of evidence assessment, which should be done by meeting the so-called Bradford-Hill criteria. Now, these Bradford-Hill criteria, without going into the details, this is something that comes from the field of epidemiology, and that define the minimal criteria that should be met in order to establish a causal link between events. So in this case, between the information blocks of an AOP. And then you end up with this bunch of questions that you need to answer, like, for example, what about the strength, the consistency, and the specificity of the MIE-AO association? Are there any alternative mechanisms that could, interfere with the AOP? Are there any uncertainties, inconsistencies, or data gaps left to be filled? Now, the second approach is a confidence assessment, which should be done by answering these so-called key questions that have been defined by the OECD themselves. And here again, same situation as for this first approach. So you need to answer, a couple of questions. So each author of a newly postulated AOP is assumed to come up with a scientifically sound answer and a very detailed answer to each of these questions. And I can assure you from my own experience, if you want to do this according to the rules, that this is quite some work. So next I would like to show you some AOP examples. So the first one is about chemical-induced skin sensitization that has been developed within the OECD. So for people not familiar with this, so skin sensitization typically consists of two stages. So the first stage is the induction stage, in which the first contact of the skin with the chemical takes place and a kind of an immunological memory, if you will, is being created. And then the second step is the elucidation phase, in which a repeated or an additional contact of the skin with the chemical occurs, and whereby the typical, clinical features become manifested. Now, a very important first step here is the penetration of the chemical into the skin, where it interacts with these local proteins, which is called haptenization. So a haptenized chemical can then induce inflammation, for example, in keratinocytes, and at the same time, it also activates these dendritic cells, as you can see here. So an activated dendritic cell then moves to the lymph nodes, where it presents its antigen to T cells, and this then serves as a kind of a trigger for the proliferation and the activation of these T cells. So what happens upon a next exposure, so a next contact of the skin with this chemical, is that these activated T cells, they directly move to the skin, where they interact with the haptenized chemical, and this then leads to the typical clinical features that we know to be associated with skin sensitization, so the rash, the redness of the skin, for instance. So a next example of an AOP has been introduced by the JRC, so the Joint Research Center of the European Commission, which is about drug-induced liver fibrosis. And in this case, as you can see here in the dark blue box, the MIE is protein alkylation and covalent protein binding. The next step is hepatocyte injury, mainly by apoptosis, which goes hand in hand with inflammation and oxidative stress. This then leads to Kupffer cell activation, and a very important event in this AOP is this expression of this transforming growth factor beta-1, because it lies at the basis for, I would say, the most important event in this AOP, namely stellate cell activation. So at the tissue level, what is then seen is the accumulation of collagen, and also in general some changes in extracellular matrix homeostasis, which is better known under the name of liver fibrosis. So another example is about drug-induced liver steatosis. Again, this has been introduced by people at the JRC, so they have been and are still quite active in the AOP field. So in this case, the MIE is the activation of a specific nuclear receptor, LXR, and this induces the transcription of a number of genes that are related to lipid metabolism, of course. Now, this not only leads to de novo fatty acid synthesis, but at the same time also to an increase of fatty acid influx from peripheral tissues, which collectively then triggers the accumulation of triglycerides. So at the organ level, what is then seen is cytoplasm displacement, nuclear distortion, and mitochondrial disruption, which then leads to the appearance of these fatty liver cells, in other words, steatosis. So this is an AOP that Bas Blober and I introduced quite recently, so it just has been published in Toxicology in Vitro. So as you probably have seen, so mainly these AOPs that have been available now, they refer to very specific toxicological endpoints. But we were surprised to see that for these very generic endpoints like basal cytotoxicity, we don't have AOPs available. So we made an attempt here in this paper to come up with such an AOP. We have defined three very important MIEs, so decompartmentalization, so for example, endoplasmatic reticulum stress, direct mitochondrial inhibition, and narcosis. So narcosis is the binding of chemicals to the phospholipid layer at the plasma membrane surface, or the actual accumulation of a chemical within the phospholipid double layer. So together this then leads to mitochondrial disruption, and this then leads to cell death by either apoptosis and necrosis. And I will be coming back to this further in this presentation, and I will exemplify how we actually can use this AOP to set up in vitro testing. Now, of course, these were just a couple of examples. So nowadays we have this AOP wiki, which was mentioned at the start of the presentation, and I think there are about 200 different AOPs that have been included in the AOP wiki. And as mentioned, they usually refer to very specific endpoints. I have listed a couple of examples here on this slide. And also, please keep in mind that this is not restricted to human toxicology. It can also refer to ecotoxicology. So, for example, acute aquatic toxicity initiated by weak acid respiratory uncoupling. So if this would be of interest to you, so I can advise you just to go to the AOP wiki. So you can see here the web address. Now this is just a slide purely informative, just to show you that there are a lot of efforts going on in the AOP field around the world. So as mentioned already, OECD has been and is still quite instrumental here. So not only they came up with this guidance on how to develop an AOP and how to evaluate an AOP, but they also made these pathway targeted case studies. Now, before the U.S. Hamner Institute of Health Sciences closed down, they were also quite active here. So again, with these pathway-targeted case studies, and focus was there on estrogen-related peroxisome proliferate-related signaling. Also in the U.S., in the Center for Alternatives to Animal Testing, where a lot of attention is being paid to so-called pathways of toxicity, POTs, and the entire collection of these POTs, which is called the human toxome. And it is specifically looked at whether omics-based information can be used in POT development. And then in Europe, I think that most of you will be familiar with this. We have this huge program called Safety Evaluation Ultimately Replacing Animal Testing, maybe better known under the acronym of SURAT. So within the SURAT program, most of the attention was paid to liver toxicity, and it was explored whether these AOPs can be used for the establishment of new in vitro test methods and also new biomarkers of toxicity. Now, I would like to elaborate a little bit more on the SURAT program because it can be used as an example on what the next step actually is. So remember, the first step was AOP development. The second step was AOP assessment or evaluation, if you will. And then the third step could be called AOP validation. And I immediately have to stress here that this should not be considered in a kind of a regulatory sense. We are talking here about scientific validation. So a couple of words about the SURAT program. So SURAT was raised in response to the European cosmetics regulation. I think you all are aware of the fact that since a couple of years we are no longer allowed in Europe to use animals for the safety testing of cosmetic products and their ingredients. So that's a testing ban. We also have the marketing ban, of course. So SURAT was a joint initiative between the European Commission on the one hand and Cosmetics Europe on the other hand. It was a project that was initiated in 2011 that was set to run over a 5-year period, so it's completed now. More than 70 research institutions were involved, and it consisted basically of 6 projects and 1 coordinating action, and maybe this is better illustrated here on the next slide. So the idea of SURAT was to come up with a kind of a strategy, if you will, to ultimately replace the use of animals for repeated dose toxicity testing in the first instance for cosmetic ingredients, but by extension to chemicals in general. So we came up with so-called building blocks that were important to build this strategy, and these were embedded in each of the different projects that made up this SURAT program. So again, I will not go into the details. If this would be of interest to you, we still have the website, which is alive. But just to mention here that the work that I will be showing to you has been performed in the context of this project, the Detective project, which was specifically addressing the identification and the investigation of human biomarkers of repeated dose toxicity. So at the start of the SURAT program, because half of the money basically came from the cosmetics industry, we wanted to know which organs are potentially targeted by cosmetic ingredients. And the major tool or the resource that we have consulted in this respect were the safety evaluation reports that are issued by the Scientific Committee for Consumer Safety, the SSCS, which is a committee that at the European level takes care of the safety evaluation of cosmetic ingredients that can be then eventually being taken up in the annexes of the cosmetics regulation. So they have an open policy, which means that they upload all their safety evaluation reports, which are called opinions, to an open website, so it's freely accessible, so you can download all of this information. And what we have done is we went through all of the safety opinions that have been published between 2000 and 2009, about 250 different ones that covered in total about 220 different cosmetic substances. And of course, in view of the scope of SURAT and the detective project, most of our attention in that respect was paid to all of the information related to repeated dose toxicity testing. So if this would be of interest to you, we have published the outcome of this screening exercise in a paper that was published in Archives of Toxicology. And just in a nutshell, we found that the liver is the most frequently targeted organ by these cosmetic ingredients, and that hepatotoxicity was mainly manifested by steatosis and cholestasis. Now, because we already had an AOP on steatosis at that time, which was discussed a couple of slides ago, we decided to first develop an AOP on cholestasis, which was not done at that time. So this was published a year later in Toxicological Sciences. You can see the reference here. So this is the AOP. So we identified a couple of MIEs, but the most important ones, especially in the context of, in the pharmaceuticals context, so to say, is the inhibition of BSAP, which is a bile salt export pump. So this leads to the accumulation of bile acids, as you can see here. So in the first instance, this then evokes a kind of a deteriorative response, which is shown here in yellow, and that is associated with, I would say, rather generic processes like inflammation, oxidative stress, cell death, all which then result at the organ level by the increased release of a number of enzymes in the serum, like, for example, ALT, AST, but specifically in the context of cholestasis, ALP, alkaline phosphatase. So that's the first kind of response. Now, at the same time, the accumulation of bile acids also drives in a kind of an adaptive, a secondary response, if you will, which you can see here in purple, and that fully relies on the activation of three nuclear receptors, which is shown here, so the PXR, FXR, and CAR. Again, without going into the details, the activation of these nuclear receptors induces a broad set of transcriptional changes, which is shown here, which altogether lead to the increased removal of bile acids and their products from the system. So as a result, there's not only increased bilirubin in the urine, but also in the serum, which in turn underlies the onset of so-called jaundice in cholestasis patients. So again, there's much more to tell about this, but we don't have time. I can only refer to our paper, of which you can see the reference here. So what we actually did then, after AOP development and assessment, according to this OECD approach, is we then wanted to check whether it actually makes sense. So this is what we have called AOP validation, which consists of two major steps. So the first step was to see or to check that we actually can reproduce what we have proposed in the AOP. And at the same time, the second step was also to investigate whether we can come up with new key events, new biomarkers, that can be included in the AOP during further AOP optimization. So we relied on in vitro experimentation here, and the in vitro system that we used for this HepRG cells, human hepatoma cells. We use a single drug, bosentan, which is used for the treatment of pulmonary hypertension. Now, bosentan, as you might know, is a very potent inhibitor of BSAP and a clinically relevant kind of an inducer of cholestasis. So this is the reason why we use this drug. So in a set of preliminary experiments, we have established three concentrations of this bosentan by an MTT test, so an IC10, IC10 divided by 10, IC10 divided by 4, which, by the way, were in line with the in vivo relevant toxic doses. We also have set up three exposure regimes, as you can see here. So 1 hour, 24 hours, and 24 hours, followed by 3 days of washout. There is, of course, this washout just to check whether the effects that are induced by bosentan are reversible or not. And the evaluation was done by using a set of transcriptomics techniques. So again, unfortunately, I don't have time to discuss the results here with you. I can imagine that some of you would be curious about it. So this actually has been embedded in a scientific publication that we just submitted. So just as a teaser, so to say, what you can see here. So this is the AOP again, and what is indicated here with the red squares are just a couple of examples of these information blocks that we were able to reproduce. But of course, the most interesting part is the new key events and the new biomarkers that we have identified. And this will be discussed in detail, of course, in this upcoming paper. So, okay, so remember that we talked about first AOP development. The second step was AOP assessment or AOP evaluation, if you will. Then the third step is AOP validation, and then the fourth step could be AOP optimization. Because it's very, very important to keep in mind that an AOP is a very flexible tool. It's an open tool. So the idea behind it is that you continuously optimize and fine-tune an AOP so before it actually becomes fit for purpose, and I will be coming back to this further on. So in terms of optimization relating to this AOP and cholestasis, what we plan to do is also some functional and in silico testing, because this for we only have performed in vitro and omics testing. Second thing on the priority list is the testing of the robustness and the application domain, if you will. So one example here is to use an other liver-based in vitro model like primary human hepatocytes. We also want to increase and test the chemical space, so not only drugs, but of course also cosmetic ingredients. Again, and I cannot emphasize this enough, in view of using this AOP, but AOPs in general for regulatory purposes, what is very important is quantification. And this can be done in a number of ways, as already explained before. So here the focus will be on the quantification of these key event relationships, for example, by establishing these concentration-response relationships. And ideally, of course, what you should also include is aspects of kinetics and exposure. So as mentioned already, an AOP is a very flexible tool. We should be continuously, refine by feeding in all the new data. But at some point, you need to decide whether your AOP is fit for purpose. And the purpose that we have in mind here for this AOP on cholestasis is, in the first instance at least, qualitative testing. So we just want to be able to have a simple yes or no answer to the question, is this chemical, with this kind of structure, potentially able to induce cholestasis or not? So what you can do here is you can set up a kind of a tier testing approach, if you will. So the first tier would be in silico testing. So just by having a look at the chemical, you could already tell or predict whether or not this chemical has the potency or the potential at least to induce cholestasis or not. So that's the first tier. The second tier would be in vitro testing. So this is exactly why I regret that the OECD gave up this distinction between intermediate steps and these key events, because this can be of valid here for this kind of weight of evidence approach. So how should you see this? So what you could do is to test a number of, something is happening with the screen. Okay, yeah. Is to set up this kind of weight of evidence approach where you have a battery of in vitro tests that are able to test either key events or intermediate steps. And basically what you should do is then run your chemical with unknown toxicological profile to this test battery, of this in vitro test battery, and every time that you have a positive result, you should just tick a box. Now, if this positive result would correspond with an intermediate step, then this would be supportive data. However, if it would correspond with an MIE or a key event, then this would be really decisive data. So this is the first thing that we have in mind. Of course, the longer-term goal is also quantitative testing, then specifically using this AOP for also establishing NOALs. But as mentioned before, you need to rely on these tools like PBPK and quantitative in vitro in vivo extrapolation. So finally, I would like to exemplify how an AOP can be used. So a first use of an AOP is chemical categorization and chemical grouping. And I think that we all know now that specific chemical structures and specific chemical groups can be associated with the induction of specific toxicological endpoints. So this can be used for the establishment of these quantitative or structure-activity relationships, which in turn can be used for read-across approaches. And the first example here comes from this AOP on chemical-induced skin sensitization, which was discussed at the start of the presentation, so in which this nucleophilic-electrophilic reaction is very important, and this is mainly seen with aldehydes, epoxides, amides, and lactams. Second example is the AOP on drug-induced liver steatosis, and remember there that the MIE was the binding to this nuclear receptor. And this is typically seen with chemicals that have phenyl rings, chloro groups, and metal moieties. The second type of application of an AOP is test development. So here the focus is not only on the MIEs, but also on the key events, which as a matter of fact can be considered as in vivo relevant biomarkers. So this can be used as the basis for the generation of new in vitro tests and by extension also in silico methods. And the example here again comes from this AOP on chemical-induced sensitization. So remember that one of the most important key events there was the activation of these dendritic cells. Well, this has served as the basis for the generation of two new tests, and I think that some of you are familiar with this. So the first is the MUST test, the myeloid 937 skin sensitization test. So here you use these human histiocytic lymphoma cells, which are incubated with a chemical under investigation. So after which you investigate the expression of CD86, which is indicative for the activation of these dendritic cells. And a very similar test is the HCLAD test, the human cell line activation test. So another cell type is used here. And in this case, you not only test CD86, but also CD54, which both are reflecting this dendritic cell activation. Well, a final or a third application, a potential application of an AOP was already exemplified before, namely tiered in vitro testing approaches. So here I will exemplify the use of this AOP on basal cytotoxicity that was introduced quite recently by Basal myself. So imagine, and I think that for some of you this is a very realistic scenario, that you would be asked to test the toxicity of a chemical, and you can only use in vitro methods or in silico methods, and there is not a single clue of information. So there is nothing you can tell based on the structure of the chemical relating to toxicity, and there is also no read-across kind of approaches available. Simply, there is no information at all. So what you can do in that case is that you can rely on this AOP of basal cytotoxicity. So remember that we have identified a number of key events there. So for example, plasma membrane damage and mitochondrial dysfunction. Well, this can be then used as the basis for this testing of a number of assays. So for example, cell plasma membrane integrity can be tested by means of this LDH test, so lactate dehydrogenase leakage test, whereas mitochondrial dysfunction can be monitored by means of an MTT test. Now, I regret a little bit that I don't have time to go into the details here, because in this paper, and you can see the reference here, we also made some recommendations on how to select the test system and also the exposure conditions. So again, in the scenario in which you would be asked to test a chemical with an unknown toxicological profile. So this can be followed by transcriptomic screening, which will give you a little bit more information about potential tissue-specific effects, or also the mechanisms that are associated with this. And this in turn will then dictate the setup of the next round of testing, which can be more focused already on tissue-specific effects. So this also will help you to rationally select a cell system, and also, like the assays that you can use for the testing of this kind of toxicity.
Mathieu, this is Graham. I just want to be aware of the time here so that we make sure.
That is actually very nice, because this is my final slide.
Oh, I'm sorry. Okay.
Last slide, just summarizing, or just a couple of examples. There are very nice AOP papers out there, but just a selection of a number of papers that have been used here as the basis for setting up this presentation. And I would be very happy to send you a PDF version of these papers. So thank you very much for listening.
Thank you, Mathieu. Jim, floor is yours.
Okay. Hello, everybody. It's a pleasure to have an opportunity to talk to you about some of the new in vitro models that are applying what Matthew's just discussed, the idea of adverse outcome pathways. And as we do that, we think about the idea that single endpoint events for cytotoxicity are really becoming passe. They just don't give us enough information. And really where we want to be going with all of this is understanding how chemicals or drugs interrupt biological processes. And that's what this diagram is showing you here. It's just giving you an overview of the idea that there are normal biological processes that occur. You have a biological input and a normal response. And when you perturb that with a chemical or drug through exposure and tissue dose, you get a biological activation, and that can result in a disruption of these normal processes. And the system can either respond through a compensatory mechanism and adapt, or it can be overwhelmed and lead to cell death and toxicity. Again, we want to focus on mechanism-based adverse events that give us information for risk assessment. And this is just an overview of what Mathieu has described quite well, just the idea that adverse outcome pathways lead us to an adverse event through key events, an initiating event, key events that occur inside the cell, and then an adverse outcome in the tissue or organism. And so this is the detailed AOP for cholestasis that I'm going to spend the rest of my time just talking about how you make an in vitro methodology or screen that can help you understand more accurately what's happening in terms of cholestasis. And in the pharmaceutical industry, there's been a huge focus on inhibition of BSEP, or the Basal export pump, and going straight from BSEP inhibition to a cholestatic event. And what we found is that as you can see, it ignores a lot of important biology that's occurring inside the cell. And as a result, while there is a correlation between the BSEP inhibition and what's happening in cholestasis, it's not perfect. And so this shows you the BSEP inhibition IC50s on the y-axis, and the line across the top is the threshold that Dawson set for the BSEP inhibition. So below that, he would have predicted cholestasis. And you can see that cholestasis, cholestatic or mixed toxicity, and hepatocellular damages, a lot of drugs do agree, or there is good correlation with BSEP inhibition in vesicles. You can see there are also along the top here many drugs that simply do not inhibit BSEP or have a very, very high IC50 and still produce adverse events in the liver. And this is another attempt to categorize that using that was done by KERS using the same data from Morgan and Dawson. And you can see here that they came up with the idea that in a BSEP IC50 in vesicles less than 25 would produce an adverse event, and in particular, that it would cause cholestatic liver toxicity. And there's a couple compounds, cyclosporin A, which is a potent inhibitor of BSEP, and also telmisartan, which although it can inhibit BSEP, they don't always result in cholestatic toxicity. So this leads us to the idea then that we can have BSEP inhibitors that are quite potent, cyclosporin and telmisartan, where we don't always have cholestatic toxicity. And we can also have the converse of that, which is molecules that have higher IC50s, but actually do correlate or do cause cholestatic toxicity. So that begs the question, how do we improve our ability to identify true molecules that cause true cholestatic liver toxicity using in vitro models? And if you go back to the way most BSEP inhibition is done, you can see that the vesicle models are very, very simple. Basically, it's a vesicle with the transporter BSEP present, and you measure the movement and inhibition of movement of probe molecules. And so all the biology is being ignored. And so what we want to do is understand the liver biology and the liver physiology to be able to make better models in vitro for predicting cholestatic toxicity. And in this case, we want to talk about the liver as being a really good example of a highly integrated organ, where you have molecules that are taken orally, absorbed in portal circulation, that end up in exposing the hepatocytes, and of course you have the formation of biocanalicular membranes and bile ducts. A very complex and interactive organ is the liver, which is responsible for most of the metabolism and movement of exogenous as well as many endogenous chemicals in the system. And one of the key things that drives this, of course, is the multitude of transporters that are present in the liver and the formation of these unique compartments, such as the biocanalicular, the bile ducts. And so we have uptake transporters, we have efflux transporters, and we have efflux transporters on the basolateral membrane and efflux transporters into the biocanalicular space. And this slide just shows the complexity then of what we expect to see in a normal functioning liver, where we have the OATs and OCT transporters representing basolateral uptake, NTCP, which brings in bile acids, and then the plethora of efflux transporters around the bile duct, as well as other transporters on the basolateral membrane. But for bile acids, which is the key to cholestatic toxicity, NTCP is the primary uptake transporter, and BSAP is the primary efflux transporter into bile under normal conditions. So to have an in vitro model that's going to predict this cholestatic toxicity, it has to be well characterized, and it has to represent all the key events, not just the initiating events that occur inside the in vivo situation. So we have to have all of these key events occurring. And those include the items listed here: energy, metabolism, fat, bile acid homeostasis, metabolism, and of course clearance. And all of the uptake and efflux transporters should be functional, present, localized correctly, and functional in order for you to understand how your chemical is affecting these processes, because those transporters control intracellular concentration of the molecules. And these are some examples of some conventional models for hepatocyte studies. And although they can provide some information, the information is limited, especially when we get to complex models such as adverse outcome pathways. And here you can see in the suspended hepatocytes, you have some of the basolateral uptake transporters present, but really you don't get expression of the efflux transporters in the bile, and of course there's no bile pockets formed. The same is true in a standard primary culture of hepatocytes. You get the hepatocytes setting down, but no bile pocket is formed. And as a result, you don't have efflux into the bile represented in the model. So in order to get a more complete model, you really have to use hepatocytes in sandwich culture. And more specifically, if you can use transporter-certified hepatocytes, which are now available in the market, you can be assured that your transporter and your functionality is present in your model. So in this model, you can see we have the hepatocytes forming tight junctions, which allows the formation of the bile pocket. And you can see the uptake transporters are functional, the efflux transporters are functional, and the basolateral efflux transporters are functional, including a very important group called the OSTs, which I'll talk about more in just a minute. Now, this model has a very good in vitro to in vivo extrapolation, good correlation. And this is just an example of one endpoint you can achieve with the sandwich cultured hepatocytes. And this is biliary clearance. And so the blue on the y-axis represents the rat in vivo. The blue on the x-axis represents the rat in vitro. And you can see the blue dots are very well correlated with both the in vitro and in vivo. And the same is true in humans. And you can see the green representing human in vitro and in vivo, and the green dots represent that good correlation. So again, just emphasizing that intracellular concentration is a key parameter for controlling or predicting what's happening inside the cell in terms of adverse events. Well, let's get back now to the idea of how do we look at bile acid homeostasis, which, as Matthew's already pointed out, is very important for controlling the end result, cholestatic toxicity. And here you can see that we have the uptake transporter represented here, NTCP, and we have the efflux transporter, BSAP, bile salt export pump, represented. So bile acids can be taken up inside the cell, and when that happens, if they can't be effluxed by BSEP, which is their primary route, then they start to build up in the cell. And when that happens, it activates FXR, which is the key trigger, the key control or focus point for bile acid homeostasis. And when FXR is activated, it activates SHIP, which downregulates or inhibits CYP7A1, which reduces bile acid synthesis, which makes sense. The hepatocyte's responding. It can't get rid of the bile acids, so it's shutting down synthesis. Another way that can happen is FXR can control the expression of FGF19, and FGF19 can also activate SHIP and cause that downregulation to occur. So when we have an intracellular concentration of bile acids that begins to increase above the comfort zone of the hepatocyte, if you will, we see activation of FXR, downregulation of bile acid synthesis, and we also see induction of BSEP, and we see induction of basolateral efflux transporters such as OST. So when we evaluate bile acid regulatory pathways, we have to use this complex integrated model. And as I said, to use transporter-certified hepatocytes gives you confidence that we're getting the same type of transporter functionality across donor groups. And so one way then to look at, we had to understand whether all of these mechanisms were in place and whether they responded in vitro the same way we'd expect them to respond in vivo. And so one way to do that is to look at changes in FGF19, which, as I showed you before, binds to this FGFR4 receptor, which inhibits SHIP and causes a downregulation in the bile acid synthesis. So then it'd be interesting in hypothesis to say, well, then if we increase the expression of FGF19, we should suppress bile acid synthesis. And conversely, if we inhibit the FGF19 receptor, we should see an increase in bile acids inside the cell. And that's what these data are showing you. So using glycolic acid as the internal probe to monitor the effects in the liver. And you can see that in the presence of FGF19, remember this is the molecule that binds to the receptor and activates SHIP, you see quite a substantial decrease in bile acid accumulation. The blue bar being the total accumulation, that's the amount of bile acid in the cell and the bile pocket, and cellular accumulation in the red, which is just in the cell itself. And then, of course, we have the changes in the media. And when we inhibit that receptor that FGF19 binds to, these parameters go back to essentially normal conditions. And that's what's summarized for you here. And what should you also notice is that the green bar is the biliary efflux marker, or the BEI, and this stays relatively constant between the treatment groups, which makes sense because the cells, BSEP's not affected, the cells are still effluxing in the bile as they should. A little additional supporting data just shows that the expression of CYP7A1 goes down. So here's the control message. Here's the reduction in the presence of FGF19. And then in the presence of inhibitor and time, it starts to increase again. So the pathway is there and functional. So now how do we look at some of the other pathways, such as FXR moderation, through other mechanisms? And these experiments were set up using 3 donors, using B-Clear technology, which is a way to measure biliary clearance. Then looking at total changes in endogenous bile acids. And those bile acids are taurocholic, glycocholic, tyrosine-deoxycholic, and GCDCA. We dosed the hepatocytes with CDCA, which is a potent FXR agonist, 72-hour exposures, and then we used a D8 labeled taurocholic acid to look at the effects on the cell system. And here's some of that data. So here we can see that over 3 donors, each bar represents a different donor. In the presence of CDCA, which is an FXR activator, we see again the decrease in the CYP7A1 messenger RNA, and we see an increase in FGF19, which is a target gene for FXR. And we also see a reduction in endogenous bile acid content. There's some variation across donors, but overall there's a nice reduction, consistent reduction in bile acid concentrations. So this is showing then that both the the ship mechanisms as well as the FXR mechanisms are in place and functional. In the presence of the CDCA, again, which is that FXR agonist, we also see a really pronounced induction of BSEP message, which you'd predict. One thing that showed up, which seems inconsistent, at least at first, is that there wasn't a concomitant increase in the biliary clearance of the TCA, the marker we were using as the probe, which at first seemed odd. But not as we began to look at the mechanisms more closely, the reason for this became a little clearer. And the reason is that the cell has been stressed by having this increase in bile acids and turning on FXR. And FXR, as I said before, is the primary trigger for controlling the homeostasis of bile acids in the liver. And what happened here is we had a huge induction of the OST beta and a nice induction of OST alpha. And these are bile acid efflux transporters on the basolateral membrane. And if you go back and look at the induction of BSEP, while there was a significant induction, it ranged from about 2.5 to 6 or 7-fold. Compare that to the increase in the OST transporter, over 50-fold in 2 of the donors and over 20-fold in the third donor. You see this is a much more pronounced effect, happens very rapidly, and allows the cell to respond. So under normal situations in the liver, then, we have uptake of bile acids from the plasma by NTCP, and normally we have efflux then from the intracellular compartment of the hepatocyte into the bile duct by BSEP. There's some efflux basolaterally, but very low. But under conditions where BSEP is inhibited and bile acids begin to accumulate, we now activate the FXR nuclear receptor, and that causes a tremendous change in OST expression on the basolateral membrane, which is a rapid release, allows a rapid release of the bile acids. It's a safety valve, if you will, to protect the liver from toxicity. So looking back at this complex biological model, then, you can see that looking at BSEP alone is not enough to predict cholestatic toxicity. And what we've been able to demonstrate in the sandwich culture human hepatocytes is that we have BSEP activity, we can activate FXR, we can induce the SHIP activation pathways, reduce the CYP7A. In other words, we've got these pathways all under control in this model, and of course OST, which makes it much more valuable for developing an in vitro screen that really predicts cholestatic toxicity. So it's likely, if you think about these pathways, that chemically induced cholestatic liver toxicity is the result of both BSEP inhibition and FXR antagonism. Because if that FXR trigger can't be pulled, the cells will not be able to initiate their compensatory response, and this can lead to toxicity. And lastly, I just want to quickly talk to you about future applications of this model in integrated organ systems. So we know that in order to be more predictive of systemic toxicity, we have to start looking at organs in combination, organs together, how one organ affects another organ. And they have to be able to be cultured in vitro under their optimal conditions. And this means that just flushing media from one compartment to another to generate a mechanical shearing force is not how it works in vivo. Ideally, we want communication between compartments to be of the drug or the chemical, its metabolites, and maybe small proteins such as cytokines. Diffusion across barrier membranes is essential for understanding these systems and to mimic the in vivo situation. And a rapid, complete circulation of complete media just doesn't do it. It's not an in vivo-like system. So one of the models we've developed over the last 3 years and now have a patent on is the multi-organ plate for looking at human tissues. And this is unique because you can use any tissue that you would like to use in this system, 3D tissue or 2D. Each compartment is isolated from the other completely. There's no net exchange of media. The only communication occurs by the simulated blood flow system, and the part of that vasculature, that simulated vascular inside the organ, is a semi-permeable membrane. And that's what allows the molecules to diffuse across, but not to have exchange in media. And this is just a two-compartment model, so an obvious extrapolation would be intestine to liver. Orally administered or orally taken in compounds enter the intestine, they're absorbed, and they enter the liver. And here we're using a 3D model from MatTech called epiintestinal. You can apply it to the lumen. You can measure diffusion across the tissue into an interstitial space, the basolateral space. You can measure uptake into the simulated blood and delivery to your next target organ. So it's really a nice way to start understanding the pharmacokinetics as well as the pharmacodynamics of your molecules. So in summary, adverse outcome pathways represent a mechanism-based approach to evaluating toxicity and that is relevant for risk assessment. In order for in vitro models to accurately predict chemical toxicity via AOP, the model must be well characterized. BSEP inhibition by vesicles does not accurately predict cholestatic liver toxicity because it leaves out the key biology, the compensatory mechanisms that are necessary to understand before you can make a prediction. The sandwich cultured human hepatocytes provide a well-characterized in vitro model for looking at cholestasis or the potential, and when you integrate that model with other organ systems, you can start building physiologically based pharmacokinetic models. And this is just to remind you that your host today was Qualyst Transporter Solutions. They're located in Research Triangle Park. They are experts in liver biology, and you can read their core expertise is here. And if you need to contact them, the information is here. And IonTox is developed a very close partnership with Qualyst. We're located in Kalamazoo, Michigan, and we focus on organ-specific toxicity screening and in vitro to in vivo extrapolation, and our contact information is here. And with that, I'd like to thank everybody for joining and turn it back over to Graham.
Thank you, Jim. Thank you, Matthew, for your very informative presentations. We have time for questions. So for all of you on the line, you can type your question into the chat box. I'll see it there. If for some reason that doesn't work for you, you can send an email directly to me. You should have my email address in the invitations that were sent out. We do have a couple of questions, and Matthew, I'm going to give you this first one. The question is, AOPs typically have a single MIE. How realistic is this?
Well, in my opinion, and I think that most of you will agree with me, it's not realistic at all. I mean, you know, like toxicology in real life, it's not a straightforward linear pathway. It's crossing of pathways, parallel pathways, and that also includes these different MIEs. Now, according to the OECD, and also for reasons of, for the sake of simplicity, so to say, the actual idea is to, have these AOPs with a single MIE, probably in view of them later merging them into a more complex model in which you have different MIEs. And this is exactly what we already have done in the cholestasis MIE. So remember, in the cholestasis AOP, so remember that we had, different MIEs there, but we focused on the BSAP inhibition because this has been very well documented, but there are many other MIEs, and this is exactly what we try to do right now. So is there also a possibility to include this in vitro testing, because this is typically focused on only, assaying or monitoring one MIE, but the more realistic case, of course, is to simultaneously monitor several MIEs at the same time. So it's not really realistic.
Thank you, Matthew. Another question for you. What could be expected from the AOP field in the coming years, and what would you recommend be prioritized?
Well, I think that we've passed the first, hype, so to say, of the AOP. So we have a lot of new AOPs that have been developed. Again, most of them from the qualitative point of view. And I think one thing that should be absolutely prioritized in the next few years, and I think that a lot of you will agree with me, is quantification, especially if we want to use AOPs for regulatory purposes, for real-time risk assessment, safety evaluation, of these chemical compounds. So I think this is absolutely number one on the priority list in the next coming years is quantification, and along with this also kinetics. I mean, we usually ignore kinetics in vitro, but this is very, very important. This may really dictate the outcome of the assay and of the toxicity testing as such. So I think this is something that also will be prioritized in general in the next few years. So quantification including kinetics.
Thank you, Mathieu, and thank you to everyone who stayed with us past the end of the hour. We still have time for just one or two more questions, if any come in. There is a question asking us if the slides will be available. We have taken a recording of today's session, and we will be posting the recording and sending you a link to that recording, and feel free to share it with your colleagues.
One thing I'd like to add, Graham, if I could, is, and Mathieu, I'd like your input too, is that species differences. So a lot of these adverse outcome pathways are set up based on knowledge that's been obtained in animals, or more specifically rodents. And as we all know, that those pathways can change. There can be subtle changes or dramatic changes as you move into humans. And so I think we need to also focus on species.
Yeah, if I may comment on this, I mean, this is actually one of the key questions of the OECD. So remember these two approaches to evaluate, to assess an AOP. So one of these questions is really focused on interspecies differences. So how specific is this AOP for species? So I'm fully with you. I mean, this is something that we should not ignore. It's really, really important.
Jim, Mathieu, thank you for those final thoughts. At this point, Daniel, I will turn the podium back to you for you to wrap up this webinar.
Thank you. This does conclude today's webinar. We thank you very much for your participation. You may disconnect your lines at this time, and have a great day.